Okay, hi. I would love to have a chance to give you a summary of the resting membrane potential, which is about the cell membrane. It's a topic about the cell membrane, but since neurons are primarily cell membrane, that's what we need to give as our background information. So if you look at a generic cell and you bring to the table this information that you already know, that would be the phospholipid bilayer right there. And you should have memorized or committed to memory the concentration gradient for sodium, which is high inside the cell, outside compared to inside. And so we would put a lot of sodium outside the cell in high concentration and low inside the cell. Then you would have a concentration gradient established for potassium that is the opposite, where you have a high inside the cell. So we'll put a bunch of red potassium particles, ions, and a low concentration for potassium outside the cell. This is pretty much the same for any cell in the body. And so I call this the generic cell. The generic cell looks like this. What we would say about the generic cell is it has osmotic equilibrium, which means the water concentration is equal on both sides of the membrane so that there's no net flow of water. That's osmotic equilibrium. And it has chemical disequilibrium. So chemical disequilibrium means that there's a high concentration and a low concentration and for ions. And I'm especially zoning in on these ions because we're going into the nervous system here, and that is these are the most important ions for the nervous system. So let's write that down. Cells have chemical disequilibrium. Disequilibrium, which has to do with the fact that there's a high concentration or low concentration. They're not equal on both sides. But they have osmotic equilibrium. And that, of course, has everything to do with the water. So the concentration of water in terms of osmoles particles is equal on both sides so that you don't get the net flow of water into or out of this cell. So that's what I would call a happy cell or a resting cell. Resting I generally put in quotes because to students they think resting is not using energy, and actually this cell is using a lot of energy. Let me show you what I mean. You kind of have to put on your 3D glasses here, and you have to imagine that there are lots and lots of potassium channels in the cell membrane, because there are. And in a neuron, there are about 40 times as many potassium channels as there are sodium channels. I just drew a bunch of potassium channels. Sodium channels are going to be fewer, but they're there. And so we'll just draw that one. Now you need to remember what you already learned about flow of ions through the channel that is specific for that particular ion. So flow for the ions through the channel is always high to low. It's always facilitated diffusion, never active transport. The other thing you need to think about that's in this generic cell, that is indeed in all the cells in the human body, is the sodium-potassium pump. And there's lots of them. I'm just going to draw one of them here for you. And I put a star there so that you could remember that yes, indeed it does use kinetic energy in the form of ATP bond energy. So now you need to put on your 3D glasses and imagine with me what is happening. Okay, for the sodium channel, what is the flow of sodium through its channel here? It can only be sodium flowing in. So all sodium channels that you have in the cell membrane are going to allow sodium to flow in. That is an electrochemical gradient because it's charged. Your book calls it that, but I always call it a concentration gradient because the flow is the same. It's down the concentration gradient, and so I always call it the concentration gradient. In your terms of your studying, you don't need to call it an electrochemical gradient. You can also call it the concentration gradient, and I won't count off for that. Okay, the problem here is that when sodium flows down its concentration gradient here through its channels, it will go from high to low until it's equal on both sides. That's the definition of facilitated diffusion. From high to low until it's equal on both sides. And then we don't have a gradient anymore. But the pump, remember what the pump does? It pumps sodium up its gradient. So let's not forget about potassium, and then we'll deal with the pump. Potassium, same thing, is going to flow high to low through its channels until it's equal on both sides. And if we didn't have the pump, you wouldn't have a gradient because it would be destroyed because of these channels, which exist in all your cells. Okay? So I'm just showing you the reality here is the pump has to work very hard. And that's why I always say the pump is the #1 energy sink in the human body, because it has to pump the ions up the gradient. Remember, it's going to pump the potassium. 2 of them are going to go in. 2 potassium in for every 3 sodiums that get pumped out. 1, 2, 3. And that is happening all the time in all your cells. So now, put on your 3D glasses and imagine that all of this is happening in the cell's environment. We've got channels for facilitated diffusion. So the sodium is flowing in, potassium is flowing out, but it's not allowed to go equal on both sides because our pump is working very hard using the energy of ATP. So let's label this as the sodium potassium ATPase, which is also the sodium potassium pump. Either one of those names is acceptable. It means the same thing. So it's using the energy to make sure that we always have a high concentration of potassium inside the cell and a high concentration of sodium outside the cell. Without the pump, there's no high and low. There's no concentration gradient. There's no potential energy. You're dead. So that book Problem about Ouabain, Oubain, is a poison of the pump. And without the pump, you lose this gradient for both sodium and potassium, and you lose the potential energy. So that's what this word means, resting membrane potential. Let me write it down for you. What are we talking about here? We're talking about this flow of ions. It's always happening, high to low, using the channels, low to high, using the pump, so that we maintain across the membrane potential energy. Okay, resting I put in quotes here because actually what this means is it means steady state. That's what resting means. Don't think it means no energy because look at this. This is all throughout the membrane. They're everywhere and they are constantly using ATP energy. So resting means steady state. For a neuron, it's not excited. It's just holding steady. Okay, not changing. But are ions flowing? Yes, all of this is happening, but it's steady. It's not changing. Membrane potential means we're seeing the form of potential energy in the ion concentration gradient. Okay, so let me write that for you. Potential energy in the form of ion gradients across the membrane. And this is all cells have a resting membrane potential, which I affectionately call, and you'll see this, RMP, for resting membrane potential. That's my own abbreviation. And all cells have a resting membrane potential, which is potential energy in the form of ion gradients across the membrane. Imagine with me that we take this generic cell and we squeeze it really, really thin so that you don't have any cytoplasm in it. Well, That's a neuron. Okay, that's what a neuron is. A neuron is basically this squeezed with no cytoplasm. And so going forward into chapter about the nervous system, the section about the nervous system, you need to be thinking about how this is happening in all of this membrane, okay? Because it is. And if you were to go inside with a probe and stick it inside the cytoplasm, anywhere inside this neuron, you would get zero charge outside and it would be negative inside. The resting membrane potential is negative inside the cell. Why? Because we have lots of channels for potassium. So look at how many positives are leaving through these red channels compared to positives coming in through the blue channels. So having a super leaky membrane to potassium makes more positive leaves because that's the gradient, the electrochemical or concentration gradient for potassium. So that's one of the main reasons why it's negative inside, because there are lots, and I'll put exclamation point, of potassium. These are called leaky channels, which means they're always open. So your generic cell has these channels, they're always open. And the other reason why it's negative inside the neuron is because of the sodium-potassium pump. The sodium-potassium pump is going to be 3 positives out for 2 positives in every time it works, which means more positives are leaving than coming in. At the steady state, it's negative inside compared to outside. That's a resting cell. It's using a lot of energy, but it's considered resting. An NAK pump, which is 3 positives out for every 2 positives in. And you learned that in your lab a couple of weeks ago. So that ratio is going to make the inside more negative than the outside. So for a neuron, the resting membrane potential is negative, and for most generic neurons, it's going to be negative 70 millivolts. Okay, that's important to memorize because going forward into the neurons, nervous system section, that number pops up lots of times. Okay, hope that was helpful.